Okay, hello everyone. We are delighted to welcome you to the quarterly HCA Town Hall. Our last meeting was in January, and over the past 2 months, there have been important updates and strategic developments that we're eager to share with you today. Today, we will cover the following topics: updates on strategic planning, Atlas updates from 2 human cell atlas bionetworks, the liver and the gut, a refresher on HCA's approach to consensus cell annotation, which is a critically important process in atlas assembly for both health and disease. And then we'll have an open discussion and Q&A where we encourage your participation. So please submit your questions and comments in the Q&A section at any time. Before we begin, we'd like to share some updates on our efforts to develop the HCA strategy for the coming years. HCA community participation plays a crucial role in shaping these strategic discussions, and today we'd like to share one such example. As we mentioned in previous town halls, HCA is launching new programs in spatial omics and disease, and these programs will be key to our ultimate goal of creating an integrated cross-tissue 4-dimensional atlas that spans all the tissues, organs, systems, and developmental stages of our body. If we go on to the next slide, yeah. At our last town hall in January, we shared plans to distribute 2 community surveys that would help us with strategic planning in those areas, and the surveys were sent out a couple of weeks ago. The first survey aims to assess the availability of spatial transcriptomic samples and spatial data to inform future planning and prioritization for generating data. And the second survey focuses on identifying global and regional priorities for disease research. If you're a member of an HCA Bionetwork or a regional network or actively participating in ongoing HCA collaborations, you should have received the survey links by email, and to those who've already taken the time to respond, a huge thank you. We'll continue collecting responses over the next several weeks, and we'll present a comprehensive analysis of the survey results at the general meeting coming up in Singapore in June. And today we'll share some preliminary feedback we've received so far. Here are some of the questions from the spatial survey. We aim to assess what types of samples are available for spatial analysis techniques and determine what fraction of the HCA community is already generating spatial data. And we currently have just under 100 responses, and here are the trends we're observing. Tissue preservation methods used, either frozen or FFP. Spatial data generated, 60% of the 100 people who applied. Tissue sampling approaches, just under half targeted. Size and source of tissues, small biopsy, larger than 76%, larger than 5 centimeters, 40%, and larger than 5 centimeters. From autopsy, 17%. So the numbers don't add up to 100 because people responded to more than one. You could respond or tick more than one box. So it's basically as a total fraction of all the people who responded. Resolution of spatial imaging analysis you're interested in, sort of about 80% was focusing on the cellular level. 43%, which is, I think, very interesting, are going into subcellular resolution. And then multi-cell and regional, sort of more coarse-grained, another 40%. Then with the disease survey, we wanted to understand which diseases are currently being studied by the HCA community members worldwide, what patient cohorts they represent, and what samples might be available for analysis. We're also interested in learning which diseases the HCA community members view as priorities for their respective bionetworks, be it tissue or organ-specific or systemic. And one of the respondents eloquently said, it would be really powerful if we could leverage the already established excellent groupings and infrastructure that came together for the healthy tissue HCA work to then tackle the generation of a disease atlas across organs. This would massively accelerate the development of potent new therapies for patients. Furthermore, bringing together researchers in an HCA disease program would add massive value, as there is no other consortium that I've worked with in this space that has had the power to drive knowledge exchange and collaborations like the HCA programs. So that is really a heartwarming response from one of the survey respondents. So if we then go on to the disease focus preliminary results, for the disease survey, we also had close to 100 responses so far, and you can see the geographic distribution on the pie chart. Most respondents currently study diseases with a high global burden, and it was interesting to see which diseases came nominated as potential future focus for HCA. Metabolic disease was most frequently mentioned, with cancer being a close second. Autoimmune diseases and a long list of infectious diseases came next, with COVID and dengue being of the highest interest. Please scan the QR codes if you'd like to participate in the surveys going forward. As I mentioned, we'll revisit this at the HCA General Meeting in June in Singapore, and thank you for advance in helping to design the future of the HCA strategy. Next, just a reminder of the HCA General Meeting, one of the most anticipated events of HCA every single year. This year, it'll take place in Singapore from June 9th to June 11th, and you can participate either in person or virtually. So the topics at the meeting are exciting. It'll feature on a few key themes shaping the future of HCA, and we count on your participation in the breakout sessions on each of those topics, and look forward to your insights and your contributions that are always essential in guiding our conversations. A few important reminders for registration. Be sure to register to attend, whether it's in person or virtual. The abstract submission is this Friday, 28th of March, so 2 more days for abstract submissions. And we particularly welcome abstracts from trainees and early career researchers, which are those who've received their degree or certificates within the past 5 years. Now, on to a couple of exciting announcements. In the coming weeks, you'll receive an email inviting you to update your profile on our new membership platform, which we're calling HCA Hub. You can think of it as an academic matchmaking platform for HCA members. It'll help you connect with others based on shared location, shared research interests, institutional affiliations, or you can search on any of those attributes, and it will also allow you to participate in surveys like the one we just talked about, engage with new HCA initiatives, and stay informed about all of the HCA events. And so as our community continues to grow, we hope that the platform helps foster new connections, but also strengthen existing ones. We have an exciting update about HCA's collaboration with UNESCO. So yesterday, HCA signed a letter of intent with UNESCO to develop a partnership to advance multiple aspects of our work, including our data ecosystem and our capacity building activities. John Randall, who's the HCA Executive Director, participated in the signing ceremony in Paris on the sidelines of a UNESCO ministerial level conference on science diplomacy. And our esteemed colleague Geneviève Almousny from Institut Curie spoke on a roundtable and highlighted HCA's contributions to science diplomacy through fostering international collaboration. Yeah, it's really a celebration and a fantastic moment. And we'll provide more updates and details of the UNESCO partnership as discussion proceeds. Thanks for everybody for the reactions. I'll hand over to Aviv now for updates on new and emerging atlases.
Thanks, Sarah. I think the UNESCO thing is just so cool. It's pretty amazing. So now we're ready to move on to the next topic on the agenda, which is the new and emerging atlases. And so first, I want to revisit some of the key points to keep us focused on the next slide. So I want to remind you what differentiates the mission of the Human Cell Atlas and our efforts from other crucial initiatives that are really focused on building tissue-specific and organ-specific atlases. So I'm kind of going to go through these items that are on the slides. First of all is our scale. We are above 100 million cells here, and also we have thousands of scientists from more than 100 countries. And this dual scale, the scale of the endeavor in terms of how many scientists participate in all the places they come from, and the scale of the data in models and information and knowledge that gets generated as a result are really distinguishing for HCA. The second is our commitment to the diversity and representative nature of the atlases so that they really represent and serve all of humanity. The third is our broad scope. And so we really aim for full body coverage, which enables cross-tissue analyses. While analyzing individual organs and tissues is extremely important, there is so much in our body that actually spans across them. And when you study systems like the immune system, or the immune system, or the adipose tissue, and so on, you actually have to understand them in the context of many different parts in the body. Sorry. The next principle is that we're flexible, but we're aligned. So on the one hand, we have a lot of flexibility to biological nuances. We realize that the world of biology and the human body is not one size fits all, but we ensure that there is crucial alignment across the atlases so that this ultimate integration into a single human cell atlas is possible. We aim to be methodologically rigorous. Methods are both created and tested by the members of our 18 Bionetworks, our standard and technology working group for the experimental methods, and our analysis working group. And then we are participatory. You don't have to have a special grant. You don't have to have been chosen in a special consortium. Anyone can be involved as long as you adhere to the values of the Human Cell Atlas, and you can come from any country, and our teams span people from many countries and from many, many different cultures. And so, we aim for the best quality, the highest quality, to have the most representation, and to ensure that our atlases are trusted globally. Now, all of those things actually often boil down also to metadata. And that brings us to the focus of today's town hall and the discussions that we will have. We place a really strong emphasis on developing rigorous metadata schemas and collecting metadata at every stage of the atlas building process. Because unless we do that, all of these lofty goals from the previous slide wouldn't really be achievable. You wouldn't get the scale because you wouldn't be able to put things together. You wouldn't be able to compare across different tissues. You wouldn't have the methodological rigor, and so on and so forth. And so our goal is to ensure that the metadata collection is comprehensive and is accessible to the researchers that contribute their source data for Atlas integration and that need to use the Atlas in their research. Now, occasionally, it may mean that we reach out to the source study authors to request additional metadata compared to what they have chosen to put out originally. And we're super grateful to people for being willing to help in completing this. And this process is really made as seamless as possible by the members of the data ecosystem team. Now, if the ecosystem team ever reaches out to you with these kinds of requests, please open their email and work with them to provide the metadata. They optimized ways to really streamline this. I know that usually when people get these questions, they're like, oh, this is going to be so much work. But we have really, thanks to the work of the ecosystem team, streamlined the metadata collection. And we made this process easy for contributors. And it actually elevates your data. It makes it a lot more likely that your study will be cited and impactful in the community. And it would make it a lot easier for you to integrate your data with that of many others and derive better insights for your research. And so metadata really plays a crucial role in the success of the Atlas. And providing it is one of your key most contributions for our shared mission. Now, there's three main categories of metadata. The first are the tier 1 data, which are the technical metadata that enables us to do cross-tissue integration. The second, or tier 2 data, are the biological metadata, and that will enable downstream biological analysis, for example, from which tissue it came. And the third level is the cell annotation metadata, and having these consistent cell labels and progressing towards a common understanding of cellular identity, of course, serves the mission of the Human Cell Atlas. And so Chloe Villani is going to talk more about the cell annotation metadata later today. On the next slide, thank you. On the next slide, this is also a reminder of the established guidelines that we have for a Bionetwork Atlas to be accepted as a version 1 HCA Atlas. So each draft atlas is presented actually to the HCA organizing committee, which is, of course, our scientists, for review, and it is evaluated on the basis of these guidelines. It has to be robust to technical factors, incorporate landmark studies, be driven by biological networks, and because we want the experts to make it, annotated with expert input. The annotations are in the cell annotation platform and have integrated data, metadata code, all of them publicly available. And only when it's like check, 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 check on all these 5 major items is the atlas considered accepted as a version 1 HCA atlas. And that means that what we put out there on behalf of the HCA is really of sufficient quality to do the work that needs to be done going forward as a scientific resource. Once the organizing committee accepts the draft atlas as a version 1, it appears on the HCA data portal, and we announce it through social media platforms. So stay tuned, because these announcements keep coming. And we're going to drop the link to the HCA data portal in the chat. Okay, so a little bit about what's coming up. By the time we're going to meet in Singapore this summer for the HCA general meeting, our goal is to have reached the midpoint of assembling version 1 of the HCA Atlas, and that would mean that we will have by then, we will have had draft atlases for 9 out of 18 biological networks: the breast, the eye, the gut, the immune system, the liver, the lung, the nervous system, oral and craniofacial, and organoids. Some of these are already released, and some of these will be forthcoming. So today, I'm super excited to welcome the coordinators of 2 of these bionetworks. The liver bionetwork recently had its version 1 atlas approved. It's going to appear on the HCA portal very soon. And we have liver bionetwork coordinators with us today. So join me in welcoming virtually Gary Bader and Alan Mullen. And then the gut bionetwork is approaching the completion of its version 1 atlas. And Jose Ordovas-Montanes, one of the coordinators of the gut bionetwork, is going to provide updates on the ongoing atlasing efforts in the gut bionetwork community. Keep the nice applauses coming. So now if we look at the assembly in the liver, I want you to join me in welcoming Gary Bader, who's going to present the recently approved version 1 liver atlas. And if you have questions for Gary, type them in the Q&A Zoom. We're going to have time for a couple of questions right after Gary's talk, and then the rest can be addressed during the discussion at the end of today's session. Gary, over to you.
Thank you very much, Aviv and Sarah and the team for inviting us. We're very excited to present the HCA Liver Atlas, which, as Aviv mentioned, was recently approved by the HCA Organizing Committee and will be on the Atlas portal soon. If you go to the next slide. This summarizes a lot of details about the Atlas, but there's 2 points that I wanted to highlight. So one is that we're creating a healthy adult atlas where we combine 7 published data sets and an additional quite a few samples of unpublished single nucleus and single cell RNA-seq data to build an integrated human liver cell atlas, which we call HLICA. And there are about 525,000 single nuclei and single cells from 110 donors in this atlas. We're also developing a disease atlas based on all the published studies that we could collect, and this will provide another few 100,000 cells and over 130 samples. And we're working on analyzing all of these together. The 7 landmark studies that we include in the liver atlas are highlighted on the left. We can move forward to the next slide. So the Liver Atlas includes data from 6 research centers, and the statistics are here: 110 donors, 156 samples. So some of the donors had multiple samples measured for them, and almost 525,000 cells after filtering. Move to the next slide. 21 experts from the Liver Bionetwork were involved in cell annotation, and these are volunteers who came together to provide their expertise to help us identify all of the different cell types that we found. And if we move to the next slide, I will talk to you a little bit about how the annotation worked, and this might also be of interest to other atlases. So we first developed an alpha annotation based on automatic cell annotation that was derived from markers that are gene expression markers that we collected from across the network. And that helped us identify major cell lineages that we wanted to split up from our atlas and focus on in zoom in annotation sessions. And so we had detailed annotation from individual postdocs who were working full-time on developing their best annotation to create a beta annotation. And then that went out to the atlas for review, where it was really easy to identify cells. We were able to do that review offline, and where it was more difficult, like for myeloid and hepatocyte lineages, we had online meetings to make decisions about how to call the cells, and that led us to get our final annotation. If we could move to the next slide. It was interesting to see how the annotation by experts helped drive our quality control, identifying clusters that we should remove and keeping others. We can move to the next slide. We also found that as we progressed through this annotation process, we got progressively more specific annotations for our final annotation, which is great to see. Next slide. So ultimately, we have called 48 cell types, which is much bigger than any other, a much larger number than any other of the studies that went into this. So by combining data, we were able to identify new biology, which is great. We think this could even go further, but these are the ones that were very clear. I think when this comes out, people want to re-annotate it. They'll likely find additional interesting things. Next. Just as an example, we were able to identify cell type markers, and in the myeloid lineage, we identified BAG3 as a novel marker and liver monocyte cell type that our myeloid team found very exciting. And there are many examples like this. I just showed one as an example. Next. In terms of diversity, we had self-reported ethnicity information from half the samples. We had roughly equal male and female distribution for the atlas, which is also great. But we did notice that by various different reasons, we didn't have genetic ancestry data for all the samples. So we were able to infer that, if you go to the next slide, by collaborating with Drew Neven, who is working on developing automatic methods for inferring genetic ancestry from the single cell RNA-seq raw data, the DNA reads that we have in our FASTQ files. And that worked really well. We were able to infer genetic ancestry from all of our samples, compare them and verify that they were matching the self-reported information we got. And also, if we had replicates from different samples, we always saw the same results, which is great. And so we can summarize the genetic ancestry components. We have some admixed samples that I didn't show, but there's all of that information is available, will be available in our metadata. Next. So next steps for the liver atlas is to make all of this information available. We already have our code available, matrices on cell by gene. We're uploading to CAP as we speak, and we're preparing a publication. But the data will be available pre-publication. Next. So with that, I'd like to thank all of the people who worked on this project. Alan and myself have been coordinating the effort. I mentioned we had lots of experts engaged in annotating the cells and helping make a lot of decisions. We have a computational team led by Gerald Kwan and myself, and lots of trainees from and staff from a number of different labs working on data assembly, quality control integration, annotation, covariate analysis, the disease map. And we'd like to thank the Lattice team for helping us with organizing our data and metadata, and the HCA portal and all the parts of the HCA for creating a framework that helps us share. And finally, this Chan Zuckerberg initiative for funding a lot of the data generation. Thanks, that's it. So are we going to take a couple of questions? Okay, so I understand I'm going to take 2 questions. You can let me know if there's more time or less time. Someone asked, is the presentation going to be on?
Gary, I can certainly help you with some questions. We have a question from Shaim Pragbakar. Congratulations on the well-structured and very logical process for lineage-specific atlas building and annotation. Can you describe your lineage-specific global strategy for projecting new data on the liver atlas?
So that's a great question. We are sharing the whole atlas as well as the lineage maps, the specific maps. We haven't tried projecting new data onto the liver atlas, but we will be doing that with the disease map. And so we will be able to share our experience with that. We will be testing different tools for that.
That's great. And one more question from Jun Ding. In disease contexts, canonical cell types often undergo significant state changes, making it difficult to map them back to their healthy counterpart. Given this challenge, how can we ensure that rare or emerging disease-associated cell populations are not overlooked during cell type annotation of your atlas?
That's a very important point. We are analyzing. We will be analyzing the disease map separately in addition to combining with the healthy atlas in a few different ways. One of those ways will be looking specifically for rare types that are not found in the main atlas. But it's an excellent point that everyone who's analyzing large atlases should be aware of, is to make sure you don't lose the rare and potentially quite important signals.
I think that's a great answer, Gary, and perhaps to remind everybody that this is version 1 atlas. So as we iterate in years to come, of course, this annotation will be updated, and we'll discuss annotation metadata later in today's town hall, and some platforms that can help give feedback on the annotation that can be helpful in the context of disease as well. Maybe, Gary, because you're so efficient with answering questions, one last question. You described an unpublished method for inferring ancestry. Tim, Trish asked, can you expand upon SCAD mix?
Yeah, so I think that will be published at some point, but this is from Drew and in Australia, and it basically detects SNPs in the raw sequencing reads from single-cell RNA-seq data. Even from 3' end sequencing, we get reads across the transcripts because of leakage, basically. So we do get quite a bit of coverage of sequence space, and we can identify SNPs on a per sample basis. And then those SNPs can be mapped to known diverse genetic ancestry genome sequencing projects to identify, to kind of infer if your sample is close to one of the other samples that's known in the database. And then that's used to infer the ancestry.
Thank you so much, Gary, again for your presentation. And also to Alan. I will invite you back for the panel discussion at the very end if time permits.
And we're moving back to the gut now, look, Chloe. So we're excited to welcome Jose, who will present the ongoing atlasing efforts on behalf of the gut Bionetworks. And like before, type your questions for Jose in the Q&A window. Jose, there we go.
Excellent. Thank you very much. Thank you very much, Aviv and Sarah, for the general introduction this morning or this afternoon, depending on where you are. And Gary already have some interesting ideas based on this. This is, I think, why these types of forums are so interesting. You can learned so much from the other bio networks as well. So today, as is the case with any real large, massive effort, I just have the privilege of presenting the work that's been really put together by the key drivers of the gut biological network. So next slide, please. This is really a fantastic team that is helping to spearhead this. So Kyle Kimmler works in our group at Boston Children's Hospital, and together with Chris Lance, who's a student at Helmholtz Munich, have really been putting together all of what I'll have the chance to share with you today. So next slide. I want to take a moment to just kind of lay out where we started and how we got to where we are today. So in 2022, Matthias Zillbauer had kind of helped to start coordinating the publication of a roadmap for the Human Gut Cell Atlas, and the references included here. This discussion was led at the HCA Vienna general meeting. And so, a plug for going to the meetings. I had the chance to be there, and this is how I personally got started on this project as well. Next slide. In 2023, the publication was finally accepted after a lot of thoughtful commentary from the assembled team. There's a lot of beautiful things that I'd like to highlight today, but really of key importance, it's just a simple reminder that the gut itself is actually quite a long tissue, and actually really many different organs itself, right? So we have the esophagus, the stomach, the small intestine, the large intestine, and then even further subdivisions within, right? There's also the axial radius that's important to keep in mind as well. And there's variants essentially that we need to deal with, both by collecting the relevant metadata and then trying to address certain challenges that have been introduced by the sample methodology that was used to acquire them. Alongside, of course, the gut is a highly environmentally responsive tissue, both in terms of its development, in terms of the relationship with the microbiome, and also with environmental factors like nutrition or pollution and toxins and so forth. And so it's really important that we use this as a starting point where we are today to really build out a more globally representative atlas over time. Next slide. So in terms of the timeline and now kind of going into last year and this year, we started to harmonize metadata and do pre-processing on existing data sets. We had at first about 10 or 11 data sets. In doing so and in starting to generate a bit of enthusiasm amongst the community, we were able to host a Bionetwork meetup in Cambridge, UK last summer. I'll touch on that in the next couple of slides, and then present kind of our progress to date at the HCA general meeting last year in Italy. We presented our kind of version 1 draft atlas to the HCA Oversight Committee, and they provided some thoughtful feedback that we're currently working on, and hoping for that version 1 approval in our next presentation to them. And then eventually what we're going to do is have a formal annotation jamboree on CAF, which we'll hear about in a little bit from Chloe. Next slide. So what the Bionetwork meetup entailed was really bringing together the Gut Atlas champions, the Gut Bionetwork members themselves, the Helmsley GCA grantees as well, and you'll see why that's been so important in a couple of slides, other large data set contributors and experts, and so forth. And the goals really here were to kind of provide a status update on where we were with current metadata collection, some of the challenges, also where we had to go forward for tier 2 metadata collection, and a variety of the other goals that were listed here as well. And really the key thing that this served is to really get a larger subset of the community involved. So next slide. The way that we've done this is we basically set up after this meeting a weekly training plan. And so we had 3 subgroups where people essentially sorted themselves into them. We have a group that meets on Mondays, M for metadata. On Tuesdays, we have the integration annotation group, and then on Wednesdays, we've had a use cases group meeting as well. And basically, they're providing key feedback, and Kyle and Chris are attending all of these meetings to be able to essentially integrate across the different groups as well. And below, you'll note the email contacts in case you would like to reach out with your contributions as well. Next slide. So what we've done is we've had the chance to bring together 29 different studies, actually 5 of them being unpublished, primarily coming from the Helmsley GCA awardees. And the number of labs currently involved in the annotation is 12. So we span a few different countries, but as you can tell, there's a large amount of work that we need to do to have a broader array of scientists helping to contribute to this overall effort. Next slide. This is the general progress report in terms of where we are. So you see that we have a few different data sets where we have duodenum, some other ones where we have small intestine, some that are ileum focused, and then other ones that have colon. A few of the data sets have the opportunity to actually span across these different tissues. And this is, of course, quite critical when you have a certain type of standardization that took place in terms of how things were collected, to be able to really look for biology over batch effects. We are looking across some different modalities, and we have gone forward with also realigning a few different data sets. And currently we're doing a reasonable job with the tier 1 metadata, and you'll see how we're kind of trying to encourage that process moving forward. And we have several data sets uploaded onto Cell by Gene and correspondingly CAP. And as the metadata comes in for other studies, continuing to push uploads there as well. Next slide. So this is just one slide. Of course, we can break this down in many different ways. In our original set of studies, we were actually lagging behind in a few different age categories. And as we know, the age being such an important driver of gut development, but something that still hasn't really been mapped at the single cell level in perhaps the detail that it should, then we try to fill this in. And so this is our latest snapshot in terms of the number of cells, donors, or samples coming from different age brackets. You can tell, of course, that things are still skewed towards the sort of 50 to 60-year-old demographic. So there's more that we can do on either end of the spectrum. And we are dealing with primarily samples coming from the ilean and particular regions of the colon as well. So there's other regions, for example, like the duodenum, that might be an interest to go into for additional versions of this atlas. Next slide. It would be remiss, of course, to not mention the names of the individuals that are kind of frequent contributors to each of the different groups. And so nothing here would be possible without them. And, real pleasure to work with everyone here on this list. Next slide. 2 points that I want to bring up in terms of the metadata and then the integration and annotation and how we've been approaching this. So metadata collection is something that is Aviv encouraged. If you get an email from us, please respond. But even after you respond, we have ways now of really following up and helping people track their progress. And so the tissue bionetworks, gut, liver, and so forth, we've been deciding on a list of different data sets that meet certain criteria. And also when we present this, being able to field additional data sets into this. Together with and in close collaboration with the HCA leadership, we've been able to agree on a set of metadata and their definitions. And then together we're trying to gather online and from contributing authors via these emails and other methods to put this into the HCA Atlas tracker. And so that Atlas tracker basically is allowing us to follow up with authors in really specific ways and be able to encourage the additional upload and tracking of the quality of the metadata that's coming in. Next slide. So the goal of collecting this metadata is not just to correct for the technical differences, but also to understand them. And I think the reason will become apparent as I go through the next few slides. Next slide. So the integration strategy that we took is, I think, probably quite similar to what others are doing, again, under the kind of general guidance of the HCA approach. So we have different feature selection that we have up front. In step 2, we are taking the count matrices and we're trying not just SCVI and Harmony, but also other methods as well. And then in step 3, we've tried various different iterations, scRNA-seq, single nucleus RNA-seq together or separate, and also split up by lineage or by core cell type. And so today I won't have the opportunity to go through everything that we've been doing, but just show essentially what we've arrived at as our final draft. Next slide. So before integration, and this is showing all the different studies, both single cell and single nucleus, colored by study, we can see that we have a few different kind of isolated islands of cells. And then after some pruning and trimming post-integration, we now have something that's a more reasonable starting point. Next slide. despite the best of our abilities, the single nucleus data was challenging to merge together with the single cell. And so towards the point of the last question that was asked, or one of the questions that was asked to Gary, we are interested in ways of now mapping the nucleus back onto this core single cell data. We eventually decided to move forward with the SCVI approach based on the integration benchmarking report that was generated by Chris and his group. And we've been working off of the cell type labels based on author cell types in order to basically assess the biological conservation and make sure that we're not overfitting. The next slide. Here are the general cell type levels. This is kind of at an intermediate resolution, just so you get a feel. We also have another layer deeper than this. Next slide. And really, if we zoom out to the cell lineage level, one key point that I wanted to share, and one thing that guided our selection of SCVI, next slide. Next, yeah, thank you, is the fact that in the Harmony integration, you can see that the epithelial cells from the small intestine and large intestine are actually being generally grouped together, whereas SCVI is able to maintain that separation. Now, it's not to say that one with the right tweaks and parameters cannot arrive at similar embeddings for both, but this gave us confidence with moving forward on the SCVI front. Next slide. And so once we have this, what we've been able to do is to estimate the variance that's contributed by different metadata covariates. And so we can see here across different categories, for example, alignment software or gut location or nuclei versus single cell. Next slide. But it's really important to remember that, of course, metadata has its own structure as well. And so Chris was able to use this metric called TILS-U, where we're basically able to say, how well does knowing what is in the x-axis allow you to guess y? And so, for example, just picking one of the examples on the right, we can see that there is some confounding between nucleus and single cell in terms of the tissue locations that have been able to be sampled. And so these are important things that we need to keep in mind. Next step. And so just flipping through the next few, just to show you the depth and the precision that we've been able to get at now with this kind of corpus of data, we can see that we have several very fine-grained cell subsets within fibroblasts, next. And also the epithelial cells, next. And the immune cells and so forth. And we can just flip to the next slide as well. And so what we've been able to do now is basically essentially start porting these things forward onto the cell annotation platform. And now we're looking forward to this community level annotation effort as well. Next slide. And this I think is now where the fun begins. And so we're very excited now that we're starting to push things onto here to be able to have a nice and lively debate on how we've done. And we really encourage everyone to please provide their feedback through this very nice platform. So next slide. And happy to take any questions or move on to discussion as is most helpful at the moment.
Thank you, Jose, for the great overview of your tour de force of you and your team. We have time for one or two questions. Maybe I ask, we have a question coming through from Shine Pribecker from Singapore, who's asking you whether they can contribute gut cancer data sets for the next iteration of your atlas.
Yeah, absolutely.
Perhaps maybe
absolutely.
you can color your response by mentioning how you may envision including disease moving forward.
Yeah, absolutely. I think that one of the key things that we need to think about now moving forward is really looking at those diseases that are of highest global burden. And so I think that gut cancer, as far as we understand, is something that is certainly emerging and continue to gain in both incidence and prevalence in many different countries. And so understanding some of the reasoning as to why, I think would be greatly aided by the contribution of these types of data sets. We have a few focused questions already just based on the ability of bringing in the large data that's been collected in inflammatory bowel disease, for example. And of course, IBD is one of the key drivers of malignant transformation in the intestine. And so being able to link those together is certainly something that would be of high interest.
That's great. Perhaps one more question before we move on to the next topic. That's coming from CU Lee, who's asking how rigorously are SCVI-based integration models typically validated and prepared for sharing, and they add more details. They found resources like the Human Lung Cell Atlas particularly valuable because their integration models are readily applicable via platforms such as SC Arches, providing a lighter and more accessible alternative to working directly with an entire data set. Are they established standards or best practice for validating or sharing such models to ensure reliability and reproducibility?
Yeah, I think you're bringing up an incredibly important point, Sayun, and it's certainly something that we are thinking about. We need to make a model that is going to be lighter than perhaps the need to download and utilize the entire data set. I think that following the standards that the Human Lung Cell Atlas set out is definitely an important starting point. But to your latter point, I don't think that there's necessarily a clear set of standards on the best way of sharing these models. And so I think this is a very active area of discussion and development at the moment. There's a lot of exciting activity in the preprint space on multiple different servers, and so I think it's something that we will need to bring in and benchmark as the HCA community grows in that direction. But I completely agree with the need that you are highlighting.
Thank you so much, Jose. And I'll invite you back to join the panel discussion at the very end. We're going to continue with our next topic, which is our approach to consensus cell annotation. And so we just had two really great presentations from the gut Bionetwork given by Gary Bader and, by Jose Ordovas-Montanes, and the liver Bionetwork given by Gary Bader. And the thing is important to remember that our ultimate goal is not just to provide you with these integrated data objects, but we're working towards generating a unified cell annotation across human tissues, right? So the goal for each of these bionetwork, for each of these organ tissues, is of course to assemble an integrated atlas of landmark studies. For the integration, we want every group to provide detailed annotation metadata based on the molecular definitions of these cells. But what's really key, and what we're going to discuss in the next few slides, is that we also need to relate the annotation metadata of the integrated atlas to legacy knowledge, to what has been published previously. And we also need a platform to gather feedback from the community, because right now, as we work towards generating these integrated atlas, these are being done by a smaller group of people. That's just to be pragmatic. But then there's an opportunity for all of you, for the entire community, to engage with the atlas assembly effort and give feedback. And so it's important to also keep in mind that version 1 atlas goes well beyond every individual tissue, goes beyond just liver, goes beyond just gut, because we need to achieve a consensus annotation for cell lineage that are also shared across tissues such as immune cells, stromal cells, vasculature, and adipose tissues, just to name a few. And so what I want to discuss in the next few slides include what needs to be done to empower the community to achieve consensus annotation, what tools and platforms are available to support annotation works, including gathering feedback from all of you, why and how do we relate integrated atlas to legacy knowledge, and how do we use consensus annotation that we will achieve in the coming 2 years for our next phase of the Human Cell Atlas. And so I'll start with why is deriving consensus annotation challenging? And I had the opportunity to present to different forums, but I want to refresh everybody's memory because this is very key to the conversation. Cell annotations can be seen as opinions that can differ between researchers. And a lot of our annotation has previously been done manually. It doesn't always scale. Very often between groups, we differ in terms of the definition of the cell type and cell states we use. It differs in terms of its molecular signature that we use, the marker genes that we use. It may differ in terms of the terminology, the vocabulary, the terms, the labels we use within the same tissue. It may even differ in terms of the precisions of terminology and vocabulary we use, depending on the type of clustering approach, for example, that we leverage. And so we asked many years ago, how can we systematically and more objectively compare cell annotation provided by domain experts to ultimately derive a consensus annotation? And that's what led to the creation of the cell annotation platform solutions that I'll expand upon over the next few slides. This platform is live as a community-driven platform that was built to create, explore, and store annotation. We provide infrastructure to accumulate, share, and analyze annotation terms with their associated molecular signatures that help interpret cellular identity. It's also a platform that will offer automated annotation service as we compile more and more consensus annotation frameworks. And we have feedback mechanisms that I'm going to describe today that are meant to encourage the researchers to converge upon a consensus nomenclature. So the first step towards building a consensus annotation is to develop cell annotation community standards, to develop a shared vocabulary. How do we go about this? We go about this by first defining in a more objective manner annotation metadata to ingest the data so we can compare and contrast how we call cells across different studies, across different groups. So we came up with 12 annotation metadata fields that are listed on the far left, including, of course, the labels, the full names you may give it, the rationale of how you label the cells, any marker genes that you would have defined using a data-driven approach, canonical or legacy markers that have been used in other modalities, such as proteins, for example. We ask you if ontology term exists and what is the closest term. If there are any synonyms, we also ask for every datasets if the datasets were annotated manually or through software. And if it's a software, we ask you how it was annotated. And we can also tailor fields for Bionetwork specific fields. And these annotation metadata give you the necessary context to more consistently, reproducibly, and accurately work with annotation as we build atlases. It also enables us to compare and contrast annotation within and across datasets, within and across tissues. And we have curators that are there to help you complete these fields, and we have a user-friendly interface to do so. I'm going to give you an example here from the Retina Bionetwork, which was the first bionetwork to get their atlas approved by the OC that I've completed these terms that you can find on the cell annotation platform. Has a different user-friendly interface with different modules that enable you to work in private space. So even if your data is not published, if you're not ready to do this in public domain, think of it as like a Google document. You can have it, you can select who has access to it to annotate your data. And when you're ready, you can publish your annotation. I'm going to show you some videos. We have different interactive user interface to help you navigate the molecular data, do pathway analysis, calculate differential gene expression and marker genes. We are launching in the coming months automated annotation service. We have frameworks to compare annotation between data sets. And ultimately, we'll be working with you as you derive consensus annotation to generate cell cards view, which I'm going to discuss in a few slides. To give you a sense, I won't give you all the demos because some of these videos are available online from previous town hall and general meetings. But for those of you who are hearing this for the first time, I want to give you a sense of how these annotation metadata come about on the interface before I tell you about feedback mechanisms. So here I'm showing you, this is just meant as a demo. You're looking at an embedding through CAP. You can actually zoom in on a particular cluster. If these cells were not annotated, you could select them like I'm selecting right now. And through this embedding, this is live computing, so you can zoom in, zoom out. You can go and click here on differential gene expression. I can do differential expression compared to the background of all cells or custom background. It's actually calculating on the far right different marker genes uniquely expressed in these cells. And as I hover over these different genes, you get a quick summary of the function of these different genes that have been reported. You can pin a gene and it's going to show you where exactly it's expressed, actually showing you that it's expressed on more cells than what I had selected. And if I hover my cursor over it, you're going to see the level of gene expression. You're also seeing on the heat map the level of gene expression of these markers compared to all other cells that do not express this marker, suggesting that this is indeed a good marker gene. And so at this point, I can create a label set. So I can give it a name. And once I give it a name, I can start entering all of these annotation metadata that I told you about. So I'm selecting this group of cells to add my label here, and then I can enter the name, and you get some prompt actually to kind of nudge you to work towards consensus also labeling. And then you can enter all of these fields. And there's more material available, and we're available to give demo as needed. And when you're done, you can add the label. Now, if you've already done this outside CAP, that is not a problem. We have different user interface so that you can simply upload your annotation directly on CAP. And you may, when we get in touch with you for every integration atlas, we ask you to upload both the individual data sets that were used to build the atlas as well as the integrated atlas. You may ask, why is that? That's because we ultimately need to relate the integrated atlas cell annotation to what has been previously published, both in terms of annotation and cell ontology. Think of it as creating the Rosetta Stone of cell annotation. We cannot simply ignore what has been published over the last several decades about cell annotation. We need the community to understand how what we're creating in terms of molecular cellular identity relates to what has been published. And that will be critical for the broader community to really embrace these new atlases. It will help and facilitate the broad deployment and usabilities of these atlases. And that's why if you have already contributed some landmark data sets, you may hear from us, from the CAP team, and engaging with us will also help increase the visibility of your data sets and ensure that the countless hours you spend at annotating your landmark data sets are accounted for when we compare your annotation to the integrated atlas. For the integrated atlas, like the gut atlas and the liver you just saw, they will be on CAP and you'll have an opportunity to give feedback on the annotation. And so what would that look like? I just told you about the 12 different annotation metadata, right? Which gives you the perfect context to start comparing these opinions of cell annotation and gather feedback. And so we can compile all of this information and go through rounds of votes together, whether we agree or we disagree. And how would this work? So if you agree, that is, if you agree with the cellular label, you agree with the marker genes, you agree with the metadata, well, on CAP, here I'm showing you one example. You could go and select a particular cell. So we're scrolling down the far left, you have all different cell subsets being labeled. I'm selecting the enteroendocrine cells. And I'm looking at marker genes here. I'm looking at these cells. I agree, I select them. And at the very bottom right, I'll be able to click on provide feedback and say, I agree. And here you can just say, I agree, or you can even add a justification if you'd like. You may actually agree with the molecular definition of the cells. That is, you agree that a group of cells represent a unique entity, but you actually may disagree with the way we've provided metadata about these cells. For example, you may disagree with the way the groups of cells were labeled, or you may recommend that we revise the metadata used. You can provide alternative in terms of chemical markers, ontology terms, categories, and full names. How would that work? So this is another example. So as you select a particular cells, you would create to gather your feedback, you would create a user account, and then this would record your feedback as follows. What you could do is you could click on refine metadata here at the very top, and then you'll be able to change or add, for example, synonyms if you wish. You could change the marker gene evidence if you wish. You could change the chemical markers if you wish, and at the very end, you can send your change, and then all of them will be compiled as a summary that will be able to be reviewed by the community, but also the Bionetwork, who can then organize meetings around these feedback mechanisms to review them and discuss them with everybody. Finally, you could disagree. You could say, I actually disagree that these groups of cells should be together and identified as a unique entity. It's conceivable that you think we have overclusters and that cells should be merged, or perhaps cells should be split. And you may consider that we've missed some rare cell subset. How would that work? So for example, here, for sake of just demonstration, we're using the T cells and examples. Here suggesting that this is too broad and this should be subclustering. So how does it work? You would, I'm just showing you that these are very broad markers. You would select these cells, and then you would go at the very bottom and provide feedback. You can disagree and you can merge or split. Here I'm splitting. And then you would tell us how many groups of cells there should be. And you provide some justification. These are just meant as an examples. You can give names for the 3 different groups, different marker genes you would recommend. And then you would send your feedback. And the goal is for every Bionetwork to gather the feedback on the integrated atlas. so that ultimately we can try to identify areas where we identified broad consensus, and then you can provide an explanation. And so once we get broad consensus, we also have ways of visualizing this on CAP so that you can also see live how this is being summarized. So for example, and this is just meant as an example, you can go and see at feedback received. So for example, we just enter some feedback for the T cells, here suggesting some split with some justification. You could also ask questions, wondering if these were annotated correctly, and so forth. You can filter based on feedback to focus on agreement or disagreement, or whether you simply, I don't know. You can look at the molecular data of the integrated atlas. Again, this is just an example. You can plot feedback at the very top of the embedding. So you could plot at whether you want to see where you find agreement of annotation, which is shown here in green. You could show where there's disagreement, and you can show where people are not exactly sure. And at the very left, you can get a sense of how many people have agreed or disagreed with your annotation. So with that, you will get for every cell labeled a summary of the feedback, the thumbs up and thumbs down. This is just meant to be a demo with some comments. All the cell annotations and authors are public, the reviews are public, and the resource will remain publicly available and timestamped. It also provides a framework to develop consensus annotation for reference atlas. So you can imagine that with such framework, you are set up to define the synonyms, right, across different cell types. So not only can we relate the integrated atlas to legacy knowledge, but among all of us, we can figure out where we ultimately agree. We've just been using different cell labels of different marker genes, and aggregating this information through these feedback mechanisms will be very key in deriving the synonyms that we're all using, both in terms of labels and marker genes, to define our cellular cell type identity. There's also an opportunity to lay over legacy knowledge, including cell ontology, based on these agreements, and update the cell ontology terms. As importantly, we can define areas where we have broad disagreement, and this gives us an opportunity to identify as a community where we should focus our effort next to resolve this conflict, and gives us a great framework to organize cell annotation jamboree in a hybrid format. You can engage the community directly through CAP. You can use the results from CAP to organize in-person meeting that can also be hybrid to discuss these results. This is just meant as a potential schema, but the goal ultimately is to be able to summarize the areas of agreement in a format such as a cell cards, in which you would get a summary about the annotation terms, the related terms, the synonyms that have been defined, the different marker genes, different data sets that have been used in literature, for example. So in summary, we now have an interactive user interface available in CAP where we can contribute cell annotation and investigate molecular signatures of annotation. We recommend for every Bionetworks that members upload all individual data sets that are used for Atlas assembly, as well as integrated Atlas. And CAP is used to compile the annotation metadata to build this Rosetta Stones of cell annotation to relate annotation across dataset and across organ system. We also offer essential functionality to review on an annotation to agree or disagree, and for colleagues to provide rationale, which is going to be key to derive a community consensus of cell subset. You can ask, how can we use consensus annotation for phase 2 of HCA? So beyond doing Atlas assembly across organ system, it's also important to be able to map the shared cell subset across organ system to build our uniform reference atlas. So for example, I also have the opportunity to help coordinate the immune Bionetwork. There are immune cells across all organ system. Some are circulating, some are resident cells. We want ultimately to have a unified cellular nomenclature across organ system for all these immune cells, which is not trivial, but the Cell Annotation platform will give us the platforms to achieve this goal. Beyond that, by compiling consensus annotation, this will empower the development of more accurate automated annotation services moving forward, because we're providing a centralized cell annotation database with refined metadata framework for individual cell annotation. And with this growing corpus of cell annotation that is paired with molecular signatures and structure rationale, we can use this as a training data for machine learning predictions. Finally, in the introductory remarks, Sarah mentioned the ongoing surveys and the next phase of HCA. We're going to be working towards 2D and a 3D atlas by leveraging different spatial measurements. And through these consensus annotation framework, we can now work together to build gene panels for different spatial platforms. And this will be especially key to align efforts across bionetworks that have shared cellular identities. I mentioned immune cells, but it applies for vasculature, stromal cells, and adipose, just to name a few, so that as we build our 2D and 3D atlas, we can also relate tissue across each other. And it will also help us annotate cellular identity more quickly in upcoming spatial data, as well as disease data sets moving forward. And so this is an opportunity for all of you, for the entire community, to get involved with the Human Cell Atlas Assembly. You all have a chance to have a word on what the final product will be. Please get involved when you're invited in annotating these atlases and giving feedback on the cell annotation platform once these atlases are posted. We have Evan Peterson has been leading the development team. Mary Fute is there to help you upload your data, answer your question about how to wrangle the data. She would be in touch to guide you to the metadata, but also is available to give demos about how to use CAP. So do get involved. Do give us feedback about the software. If there's specific features you would like. We would like to hear from you. We're there to help you. So help us help you. So with that, I will stop sharing for a moment and see if there's any questions for CAP, but also invite the panelists, including Jose, Gary, Alan, to join me back. And Sarah is here as well, so we can take on more general questions. And there was an earlier questions about disease, which I think might have been answered. Sarah, you've answered
yeah, I answered a couple of them.
Would you like to express? So maybe I will mention the questions because I think that's going to be a question of interest to many. So that was from Benjamin Tuwazi. Each region and sub-regions across the world has their own disease burden. How do we make sure that all diseases are captured? Sarah, would you like to expand on your answer?
So what I said was that basically there are, I don't know, 25,000 registered diseases, so we can't address all diseases in all regions, but what we can do is survey the community and find out what the opinions are on priority disease areas in different regions around the world, and then have a conversation about prioritization of the HCA community in terms of addressing kind of the most urgent sort of and most interesting, scientifically kind of interesting diseases around the world.
Yeah, and thank you, Sarah, for this clarification. And perhaps I can add to Benjamin, our goal here, to add to Sarah's comment, is not to map every single disease out there, but to be complementary to ongoing efforts around the world that are already studying disease through different networks. We realize that some diseases are perhaps not as studies, and we also have a unique opportunity to connect members around the world studying the same disease. And so through these surveys, we can figure out new priorities based on availabilities, but of course, a number of groups studying the same disease around the world. And that's why answering this survey is extremely important, because you can help us decide which priorities to focus on by providing information about what you're currently working on. And as we move forward, this is not going to be static. We'll update the information as we go and update priorities, but we need to start somewhere. Perhaps around this, so Jose mentioned a few diseases you'd like to study. I don't know if Alan and Gary would like to add any comments about also upcoming disease data sets you're envisioning adding in the next iteration.
Go ahead, Alan.
Yeah, I think that within the liver, certainly one of the categories that you included earlier in terms of metabolic disease, so metabolic dysfunction associated steatotic liver disease, which is really the sort of the major problem causing chronic injury, or probably the most common reason why we see patients with liver injury, is certainly of a lot of interest. I think other areas, we know that fibrosis is an end stage injury, and a lot of groups have been collecting samples around those areas. And certainly Toronto's group has been leading a lot of work in the area of a condition where we don't even have a treatment for chronic liver disease. It's a fibrosis called primary sclerosing cholangitis. So I feel like those are some of the areas where we're seeing the most availability of samples and interest, both in common diseases, but also those that are very narrow, but don't have options for treatment.
And also liver cancers, like hepatocellular carcinoma.
Oh, absolutely.
Yeah. Jose, is there anything else you'd like to add? I know you're also quite involved with Inflammatory Bowel Disease Network. Do you want to expand a little bit on this?
Yeah, I think the important thing to remember, and especially in tissues like the intestine, is that many of the different samples that we're already working with in terms of our first draft could potentially have come from some of these IBD studies, but we've been quite rigorous in terms of really trying to select things that are as healthy as possible. And so actually in the draft that I presented today, we have had the opportunity to essentially put off to the side for the time being those non-inflamed adjacent biopsies that can be taken, and of course the overtly inflamed biopsies as well. So that gives us the chance quite quickly to, from those same studies that have been processed and treated in the same way, to then bring them back and help to interpret them within the context of our atlas. I saw a question that in the chat before that I think is very important to address, which has to do with, how can we be certain that we are able to then confidently map or understand a disease-associated cell type if the main focus is currently one of health, right? Or as healthy as possible when it comes to the atlas building efforts. And there, I think it's one where we might find correspondence related to some of the underlying structure of that cell, but actually not finding a perfect match is really the important feature, right, of having the carefully curated controlled data. That's how you start to understand, oh, I might have a certain new feature in disease that has exceeded, the normal parameters that help us to define that as a discrete different cell subset. So yeah, I think I view that as an opportunity, if anything.
That's an excellent point, Jose. Alan, Gary, Jose, you both presented, well, the three of you presented for your group the first version of your atlas that are being finalized. Is there anything you'd like to recommend to people that are currently lesson learned from what you've done that have really improved your atlas assembly that you would like to recommend to other groups that are considering atlas assembly right now? It looks like Jose, you were nodding. You'd like to go first?
Nodding doesn't necessarily mean going first. I was agreeing with what you were saying, but I do have a few thoughts that I can share while Gary and Alan come up with better answers than mine. But I think that what we have really learned is the importance of trying to come together as a group to really help kickstart things, gain that familiarity across people that may only know each other from Slack conversations, for example. And then from there, we really have helped to catalyze and energize the groups that continue to push on between those times. And I think that dividing things up into discrete goals, right? Not just saying, oh, we're meeting as a group, right, to talk about stuff, saying this is about metadata, this is about integration, this is about use cases, and then having those champions, Kyle and Chris in particular, that help to unify those different conversations. I think that when we set in that structure into place, suddenly then things were really moving forward. And so it's much more than just, pasting together a list of data sets and saying, let's move forward with this. A lot of it has to do with making sure that we have the right structures in place. And, we're constantly in communication as well with you and with other HCA leadership about, the latest and greatest in terms of how we should be evaluating what we're putting together.
I think that's a great also opportunity to remind the listeners of the town hall of a call that Aviv made earlier on, which is we may be reaching out to you if you contributed a landmark data set. And it's really an opportunity to help elevate your studies when we do so by providing this metadata so that we can properly integrate it. And we have a whole team, including Kyle, that is part of Jose's team, who spent a lot of time in trying to think about how to ease the pain of getting this metadata. And so it's not as big of a lift as what you may think when we reach out. And that really eases the process for downstream atlas integration and ensures that we really capture properly the biology that landmark studies have previously reported. Alan, Gary, any other thoughts?
Yeah, I could go. We had a lot of things that worked quite well, but we also, looking back, there were certain things that slowed us down. I think that if we're starting an Atlas project now, we would have had a more priority on organizing our compute platform centrally for everybody, because we decided to align all of our data from raw FASTQ files with the same cell ranger pipeline. Not everyone's doing that, and it's not clear that that's how much value that gets us yet. It's like an open question still, but it did require a lot of computing and downloading data and wrangling data. And we're very fortunate to have that Lattice team helping us to provide like cloud-based, systems to store things, and also help collecting metadata from individual groups. I think that overall, we're quite happy with things, and I think that it would be good for us to write that up somehow. I don't know how the atlases are going to be sharing their experiences in any formal way other than through these presentations, but we might think about, discussing that. I know there's also Malta's team is sharing information in regular calls through trainees that are, working on the Atlas projects and talking to each other. So that's been great. Yeah, it would be good to share that information somehow. And
Thank you, Gary.
of course now with
I mean, I think Malta will, have a sort of overarching, a further kind of overarching guidelines papers, but I'm not sure it'll include the kind of thing that you mentioned, Gary. It's quite detailed. Yeah.
Right, and perhaps Sarah, I'll invite you also to reflect on some of these comments for Atlas integration, but perhaps to add to Sarah's comment, Gary, I think for every Atlas being reported, we invite the Bionetwork to provide a summary on the HCA portal about how you've built the Atlas and how you got to your data object, what type of metrics you used to qualify and quantify your integration and decide that this was the most optimal integrated object for this data freeze. And so there's also an opportunity to provide some level of details on the HCA data portal for every Atlas, but also through the code being released, because we ask for sake of reproducibility and transparency that the code for every version of the Atlas be released. It really helps moving forward as we think about the next phase. As it was pointed out in the introductory remarks for Sarah and Aviv, this is just version 1 of the Atlas. We're going to continue iterating. And we also need to connect cells across organ system. And that's why we really push for this data structure, metadata, and transparency. So these lessons learned here are incredibly important. Before I move to Sarah, who is involved in many atlases and can give us a broader overview of her thoughts. Alan, any other thoughts you'd like to add for liver and the biology?
Yeah, I think the other thing that I know Gary and Gerald worked really hard was to try to get groups together to study, like to talk about individual cell types. And it sounds like the group did a really nice job of actually getting people physically together. We really managed to figure out how to do that, but I think by dividing into areas of expertise like the myeloid section of cells, the hepatocytes, and trying to get those groups together to be able to at least talk both in person and then going through the objects and looking at the cells ahead of time made a huge difference in terms of kind of finalizing some of these annotations.
That's really great. Sarah, through your leadership, you've been involved with several Atlas Assembly with tremendous lesson learned. Any thoughts you'd like to share for what we've done and where we're going next?
Yeah, I think, now that we've released 4, basically, so lung, brain, organoids, and eye, I think we're kind of we've seen sort of different models. So there's been some uniformity and some kind of sharing across pipelines and optimization and so on. But there's also been, just because of the evolution of the different bionetworks, as you're saying, Chloe, there have also been kind of idiosyncrasies or specific ways of doing things in the 4 different ones that have come so far. I really appreciated kind of the insights into what's on the horizon from the liver and gut. And my feeling is now, we've got kidney. I mean, development is a slightly special one, sort of embryonic and fetal stages, but for the major organs, heart, kidney, and so on, I think there will be a more sort of uniform pipeline, like we've heard from liver and gut just now. I should say there are 2 questions, so we only have another few minutes left.
I will tackle them.
I would 2 questions in the Q&A.
I was going to tackle them since they were annotation, but I want to give you the last word.
Right.
I didn't notice. And perhaps thank you, Sarah, to add to Sarah's point, as it was perhaps illustrated best by today's presentation from the gut and the liver Bionetwork, there's not a one-size-fits-all solutions for Atlas Assembly. The important thing for us is that the groups are transparent about how they've assembled the Atlas, that they test more than one integration methods. As you've seen, they are providing quantitative and qualitative metrics to justify the integration and then working together towards annotation. So it's not about a one-size-fits-all solution. It's about tailoring the integration approach to every organ system, which are defined by unique biological entities, hence the tailoring of the approach. To Sarah's point, there are 2 questions related to annotation in the chat that I'm going to briefly answer from Mohamed Tehrani. In annotating cells, do you use physical, morphological, chemical measurements of the cells? Can we define such parameters that are coming to a larger majority of cells? That's an excellent question, Mohammed. For the first version of the Atlas, a lot of the annotation is being done through the lens of molecular definitions, since the majority of our data point across organ systems is in the RNA space. It is not, and so that's shared across all organ systems. So for this first iteration, the common ground for all Bionetwork, all organs, is this molecular definitions. But on top of the molecular definitions, as we gather more information about the cells, we can layer these features, which can help find cellular identity. So as this information becomes available, that certainly can be added. Jun Ding asked, for cell type annotation within the HCA framework, are there current efforts or future plans to incorporate active learning strategies to optimize manual annotation, that is, AI/ML methods to use to flag the most uncertain or challenging cell population where expert input is most needed, so that manual curation efforts are prioritized currently? Not all annotation carry equal uncertainty or impact. That's an excellent question, June. There are several automated annotation services that are currently available. What we're doing for the cell annotation platform is actually encouraging an initial phase of some level of manual curation so that we can create some strong foundations for annotation and drive a consensus. And at least that is on the roadmap for the cell annotation platform, to leverage this consensus annotation to provide updated service for annotation service. We are also currently integrating different automated annotations package that have been published by the literature, by the community, so that you can use a popular voting approach to select annotation. And so that's definitely part of the roadmap, but this is a shout out for contributing to consistent annotation, because if you all contribute to working towards consistent annotation, these automated annotation service will become more and more accurate in years to come. So with that, Sarah, I will turn to you for concluding remarks. I will share my screen again.
Thank you, Chloe, and thank you to all the panel participants. Alan, Gary, Jose, I think it's been a really exciting window into sort of where we stand in the Human Cell Atlas project right now, having these integrated reference objects, training kind of on refining the annotations, iterating on refining the annotations, which is going to be amazing for foundation model training, for cross-tissue integration, ultimately. So it's a real pleasure and an honor. We really appreciate your partnership and everybody who's been on this call, and hope to see you in June or at another HCA meeting in the near future. Thank you, and thank you to all our partners.